Okay, let's go ahead and get started. Welcome back. For those of you that this is your first one out of the three, welcome. For those of you that have been with all three, this is it. One last to go, and then we get to go have some fun and drinks or whatever. So this presentation has to do with the new FDA draft document that was released last year. And this was very big news in the biocompatibility arena. In fact, to give you a little bit of background on how it came out, is every year as an ISO 10993 working group, we meet together and we go over the standards. Last year in April, we were in Milan, we were in a place called Pavia, Italy, and and the FDA couldn't make it because they were sequestered at the time. And so all of us were sitting in a room, actually there's like 3 or 4 rooms, and all of a sudden someone raised their hand and said, the FDA just released their new draft document. And we went, what? Because we didn't even know they were writing one. And so they kind of closed down the meeting so we could all read the new draft document and start talking about it. So the reason why this was big news was because the current document that we use for biocompatibility is the G95, the FDA G95. And that 95 means the year it was made, which was 1995, which was a long time ago. And so that document is basically not really used very well right now. So the FDA released this new document, and so it's great because we actually get an up-to-date insight on what the FDA thinks as far as biocompatibility. And it's something that we've referenced to the FDA, even though it's in draft. We still reference it in risk assessments and things like that because it's the most current thinking. So this is the cover page. If you haven't read it, if you want to be able to, go to sleep tonight, you can start reading it. It's that enthralling. But this is the link in case you want to go there and read it. And if you don't want to read it, I read it and I went through it and I broke it down into 3 categories that I kind of want to share with you. This kind of gives you what you need to know out of the document, and so you don't have to spend so much time going through it and combing through it. But the 3 categories that I broke it down into, the first one is a summary of ideas. And what this means is they're ideas that we have seen in the last 5 or 6 years from the FDA, but we've never had them written down anywhere. So it's kind of been feedback we've seen from the FDA, but not like in an official document. So now it's been put into an official document. The next thing is actually the FDA gives us ideas to help justify out of biocompatibility. So it really is what it is they got to a point where they're thinking, look, we need to give people guidance on what to think about when they're justifying out of tests. So they give us some ideas of ways to get out of doing biocompatibility. The last category are things that we have never seen before. There are things that I have never seen the FDA ask for, and now they're all of a sudden putting into this new document. So it's going to be interesting to see the impact going forward. Before we go into it, to give you an update on where the document is, so the document was headed by Jennifer Good from the FDA, and she asked for 6 months to be able to receive comments on the document. She received something like 600 and something comments. And so she's still going through those comments and discussing them. So the last I talked to her was a couple months ago, and she says they're still going through them. So I asked her if there was any kind of idea when we would see a new draft or an updated document, and she says it should be next year sometime. That's kind of vague, but that's what I got. So we're going to go ahead and go through the draft now as it stands, and just keep in mind that it's possible that some of these would change depending on the comments received to the FDA. So the first thing we're going to talk about is these summary of ideas. So remember, these are things that we've seen in responses from submissions, but we haven't seen them written down until now. The first one is this document incorporates new ideas regarding toxic chemicals, specifically colors, previously not addressed in G95. And if you were here in my last talk, we talked about colorants. And right now the FDA has a huge concern with colorants. So if you are going to add a colorant to your device, please think about it, because the cost it's going to take to be able to show that colorant's safe is going to be quite large. So if it's beneficial enough to overcome that initial cost, then we can do it. But think about how beneficial it is. If marketing just wants to make it purple, go back to marketing and say, do you really want this purple? What's the benefit of making it purple? Is it going to outweigh my cost and time in testing? This is one that we've seen, and I've actually really recommended. It's the sponsors are advised to discuss testing plan with the FDA before initiating testing. And I recommend this if you're looking at doing something that might be a little bit more risky. Okay, so if you're looking at justifying out of testing, if you're looking at maybe doing less testing, or if you're not just checkboxing, what we can do is we can get together a test plan. Okay, so we can talk about your product, we can talk about its intended use, its materials, and we can get a proactive test plan together with the justifications in place, submit that to the FDA, and then they'll get back to you and respond to let you know if it's a correct plan or not. There are a couple things to think about. First one is there are a couple of different versions of this. There's an informal meeting. Okay, the informal meeting, which we've had before, is where you actually get to just talk with the FDA reviewers and their experts and discuss their plan or the biocompatibility. But whatever's said in that meeting is not considered official, but you still get good insights on their thought process. Then you can do an official pre-sub or Q-sub with them, which is this official recorded meeting where the minutes are recorded, and they have told us that any decisions in that meeting, as long as you start testing within 6 months, that they'll hold to those decisions. So in this case, you can actually sit down, go through each test procedure with them, talk about how you're going to prep the sample, how you're going to extract the samples, what testing you're going to do, how you're going to assess each concern, get their feedback, and know that when you start your testing, that as long as all the testing goes well, that you should have a valid result in your submission. The next thing is full tests are needed for FDA submission. We still have some people who are just providing summaries, and actually, to be honest, I've seen some summaries still get accepted. But the FDA is starting to ask for full test reports because they want to see how the sample is prepared, how much of the material is added. They really want to get into details of the test themselves. This is also something that you'll see throughout, like kind of a thread throughout the whole document, which is the FDA does not approve single materials. So we have a lot of devices that are made out of FDA or biocompatible biomaterials. And so the FDA, a lot of people will try to say, look, I'm using polypropylene, I'm using PEEK, they all have medical grade certifications, I don't need to do any biocomp. The FDA does not approve just materials because there's processing that goes into making the device and you're sterilizing it, which can have an impact in biocompatibility. So when you're looking at justifying out of a test, you can't just look at materials, but you also have to look at what you're doing to those materials as part of your justification. This is one that not everybody has, but if you have a specific target organ for a device, we have to do toxicity on that specific target organ. But this is mostly applied to neurotoxicity. So if you have a neuro device, we have to do neurotoxicity, which is a complicated custom study that we can do for you. We've done it in the past, but most devices don't have a target organ that they target. Some of them that do have target organs, like hearts, we can do toxicity with vascular toxicity and not have to worry about heart specific. Really, this has to do with neurotoxicity. The next one kind of is that thread again, you'll see, master files are not sufficient for submission. We have people that say, hey, my NuSil silicones has a master file with the FDA, so I don't have to do any biocomp. If it has a master file, that's great. We can use that, but once again, you're doing processes to it, you're doing sterilization to it. What did the master file have done to that material? Was it sterilized or was it a non-sterile product? There are some questions that we have to do to be able to assess the biocompatibility, even if it has a master file. The testing should be conducted GLP. So for those of you that don't know, GLP stands for Good Laboratory Practice. A GLP test and a non-GLP test are ran exactly the same. There's no difference on my part how they're ran. The difference comes with how they're regulated. So we have an independent QA person that logs them in, they audit the testing throughout, they review their study, so there's just more quality controls built in. So the FDA likes those quality controls, so they require submission to be GLP. For biocompatibility tests, this is a very small portion of the cost of the testing. So we always recommend it be done GLP. There's really no reason not to do it GLP for biocompatibility. Let me take that back. If you're just screening the product and you're not submitting that data to the FDA, you can do it non-GLP because the testing is the same. But if you're submitting to the FDA, you want to do it GLP. Okay, this is one that kind of shocked us, but we've seen some of the feedback from the FDA on it, which is for permanent contacting devices like implants, you should either perform or need to justify chronic toxicity and carcinogenicity testing. In the past, these two test categories were kind of just forgotten. Not many people perform chronic toxicity or carcinogenicity testing. Now, the FDA wants you to evaluate the need to perform those two tests. Honestly, the carcinogenicity test is easy to justify because you look at your materials and the cancer risks of the materials and the processing. The only thing in a medical device that has a high risk for carcinogenicity are colorants and some chemicals. So those are why the colorants are a big concern from the FDA. But we have to be able to address those, and usually it's just a written justification why chronic toxicity and carcinogenicity are not needed. The other thing that they gave us in this document that I actually love, which is they gave us guidance on how to make a coupon device. For those of you that don't know, a coupon device is a device that represents the final product. For biocompatibility, I don't need to test a final device. I need a device with the same materials and the same proportions that undergo the same processing. But I don't need a functional device. And so a lot of times we test on coupons, which is a different form of the device but represents it through the materials and processing. The FDA has given us guidance on how to make a coupon, how they'll accept a coupon device. And this is beneficial for devices that are very small or very large that cost a lot of money. So I don't need that huge device, or I don't need that small device. I can make a bigger device that represents it. If you have a device you think would benefit from a coupon, talk to us. We'll help you make sure that your coupon meets this guidance from the new draft document. This is another one that makes me smile. It actually says in there that the chart in ISO 10993 and in this new draft document is not intended to be a checklist, okay? The FDA does not want you just to go down and check, do all the tests. I still have people who do that because they think it's safe and they'd rather just do that than risk anything. But the FDA really does want you to evaluate the need of the testing on your device and justify why you're doing those tests and why you're not doing other tests. They want you to be scientific approach and not just throw the phone book at it, so to speak. Okay, this is one that once again we've seen happen, and the FDA is now putting it in this document, which is failure options. So we have lots of devices that could fail during use. A balloon is the most common. So a balloon that might have fluid running through it to blow it up, if that balloon bursts, now you have all the internal components that aren't intended to have patient contact, now would because that fluid goes into the body. So you have to analyze your device and think of failure options of that device, and if it fails, would it expose portions of the device that not normally would be intended to be exposed? There's always a distinction between a failure scenario and an insane scenario of failure, right? I've had people that say, well, there's an earthquake that could tumble this thing over, and then this portion of it, I mean, no, we're not talking about that, right? We're talking about a realistic failure that could happen during the procedure that would expose portions of the device that aren't normally exposed. Another example is we had a drill guide that's put on the bone that the surgeon will drill through to make the marks in the bone. That drill guide is supposed to have very limited contact with the patient. But sometimes a patient or a doctor would drill up against the drill guide and shed some of that material into the wound. And we all know doctors aren't going to spend the time to pick out every single one of those little materials. So now that sponsor had to evaluate permanent implant for that drill guide instead of just short-term contact. Another example of a failure scenario. Mass extract volumes should be only considered if surface area cannot be determined or if mass is worst case. This is out of ISO 10993-12. We're given two options to determine the extraction ratios for a device. We can either do it by its surface area or by its weight. Surface area by large is worst case compared to weight. So if you're going to submit your device to the FDA using mass ratio for an extract, the FDA is going to ask you to justify why. So use surface area whenever possible. The exceptions to that would be if it's an absorbent sample, like a sponge, or if it's a bioabsorbable sample, or if it's a powder. Those things are very difficult to get surface area for, so we can do weight for those. Almost everything else we should use weight for. This is other one for prolonged contact devices, for devices with longer than 24-hour contact with the body. They want to extract for 50 degrees C. instead of 37. And that's because we can only extract for 72 hours at 37 degrees. So if you have a long-term contact with the body, the FDA wants to see what happens over a long period of time. So they want you to elevate the extraction fluid to 50 degree, or temperature to 50 degrees C. I actually recommend doing 50 degrees C's for anything. And that's because my philosophy is I'd rather not have to explain something to the FDA unless I have to. So if your device is made out of material that can handle 50 degrees C, just extract at 50 degrees C. Then you'll never get that question of why you extracted at 37. Or you have to think about explaining why you extracted at 37 degrees. We cannot manipulate the extracts. Okay, so we can't centrifuge, we can't filter, and this is hard when we have devices that may shed particulates, especially when we cut them up into pieces like we do when we extract them. So just keep that in mind. We can't, or if we do manipulate it, we have to justify why it's appropriate. For your cytotoxicity test, it should be extracted using 5% calf serum. Some labs use 10% calf serum. Some labs still use 10% calf serum. So check with your lab. Make sure they're using 5% calf serum because that's what the FDA is preferring. For blood contacting devices, regardless of duration of contact with the blood, the FDA is requiring hemolysis, complement, and thrombogenicity tests. Okay? This is something we've seen very commonly. So it doesn't matter, you can follow Table 1 out of ISO 10993-4 to try to interpret what tests are needed. The FDA does not recognize that. They'll just have you do these 3 tests no matter of your duration of contact with the circulatory system. Intramuscular implants are preferred over subcutaneous because of increased sensitivity. So if you're going to do an implant, make sure it's intramuscular. Okay, so the next one is justification ideas. These are ideas the FDA gives us to justify out of doing biocompatibility tests, which can be really helpful, right? The first one, which I think most people should use more often, which is that you can weigh the benefit of a device over its risk of the patient, right? So a good example of this is chemotherapy drugs, right? Chemotherapy drugs are extremely toxic, but the benefit to a cancer patient outweighs their toxicity. If your device is going to prolong someone's life or is going to save someone's life, then you can be a little bit more toxic. You can have a little bit more impact in the biocompatibility tests and be able to justify those test failures, or even looking at carcinogenicity and toxicity a little bit differently because of the benefit to the patient. This is also one that makes me smile. Reviewers are encouraged to apply quote-unquote scientific judgment when reviewing alternative data for animal tests. And this is great because I just spent this morning talking about how we're going to look at alternatives to animal tests using in vitro alternatives. And we have the chemistry that we can use to help justify biocompatibility. All these alternatives to animal tests. And the FDA, the people on the top, are telling their reviewers, look at alternatives to animal tests. Encourage them to, clap, yay, to encourage them to look at alternatives to animal tests. And we're actually seeing the reviewers now get that message. It was a long path, but we're starting to see them now look at alternatives to animal tests as a viable option. Literature can be used out of systemic toxicity tests, but the correct data point has to be used. If you need help with this, let us know, but you can actually go into the literature and look for toxicity tests that have been ran on your material, as long as you're looking at the same toxicity endpoints. So if you're trying to justify out of subchronic toxicity, you have to find subchronic endpoints. If you want to justify out of subacute toxicity, you have to look at subacute endpoints. You also have to look at the data to show that the same histopathology, clinical chemistry was performed, but we can use that data to justify out of testing as long as that data represents the final device. We can help you with that if you need help. Yes? We do it all the time. We actually write risk assessments for materials for out of chronic tox and subchronic tox based on data we find. Okay, so the next one with justification is biocompatibility testing and or adequate chemical characterization in conjunction with supplementary biocompatibility tests may be acceptable to minimize animal testing. It's a lot of words, it's a quote unquote, but basically what that means is that adequate chemistry can help us justify out of animal tests, especially with the genotoxicity and chronic toxicity, okay? We can use chemistry to justify out of. This next one gives you kind of an idea of what the FDA looks for to justify out of testing using a predicate device. A lot of people say my competitors or my previous device, they have this, biocompatibility, mine's exactly the same, so I don't need to do anything. Well, the FDA says if the intended use, physical form, formulation, processing, component interaction, and storage conditions are the same as a predicate sample, no/limited testing would be required. So it gives you what the FDA looks for justifying. For me, that's going to be hard to get for my competitor product. But if you have your own product that's on the market that has a lot of these same endpoints, then we can use that to justify out of biocompatibility. And we've done so very successfully. This next one is also one that we've used successfully. In fact, I try to get people, when they're doing their functionality tests, to call me first, okay? Because a lot of times the functionality is done by somebody else, and then the preclinical testing is done by somebody, in another department, and they've wasted some of their endpoints in their functionality tests for biocompatibility. We can put endpoints in functionality. We can put hemocompatibility. We can put implantation data. We can put systemic toxicity data in functionality tests. If you call me, I'll give you my protocol. I won't even run the tests. I'll let you have what we look for, and so you can put it in your functionality protocol, so therefore we can use that data to justify out of the testing later on. As long as the product doesn't change, then we can use that data, okay? Now, the last category we're going to cover is possible impacts. Remember, these are things that we have never seen before that can be a little scary when we're looking at the biocompatibility. Devices that have multiple types of exposure should consider testing from both categories of your device. And the question we asked about here is we've seen this some, but we want to kind of understand a little bit more about what they're trying to say. For example, if you have a device that has skin contact and it's inside the body, do we have to separate them out, or can't we include them together because in the more sensitive test system? Or do we really have to start testing some portions of the device that contact the skin differently, separate? Because now we're just increasing the amount of tests that we're doing. It makes sense, I think, and we'll get into that in a minute. I think they're getting to like a stent in its delivery device. They don't want to test the delivery device with the stent. They want to separate it. But it's a little too vague in my opinion. There's too many avenues that the reviewers can go down that I don't like. So that gets into the next one, which is the devices that include components with different lengths of contact, like an implant and its delivery system, conduct extraction tests on the components separately, okay? So no more running the stent with the delivery device on the big three. We have to separate them out. And so we just want to, once again, make sure about that. This next one's probably the one that scares me the most, and it scares me for orthopedic devices, because it says implants, devices in contact with cardiovascular, lymphatic, or cerebral fluid, or labeled as non-pyrogenic, need to perform the LAL test along with rabbit pyrogen. And the reason it scares me is because that comma up there after the implant suggests that all devices that are considered implants need to do lot-to-lot LAL tests. And right now, only devices that are in contact with cerebral fluid, lymphatics, cardiovascular, or is labeled as non-pyrogenic have to do those tests, or the LAL test. So I have a couple of friends that are ex-FDA, and they said that when they were in the FDA, they had this discussion that implants should have to do LALs because of cleanliness. They want to make sure they're clean, and an LAL is a way to kind of help show that they're clean. I think the burden on the device manufacturers, on the implants, orthopedic companies would be great, because it's a lot-to-lot release test that they have to perform. So we asked to see if that's true, but we'll hear back from them as far as if they're going to enforce that whenever the new document comes out. It's a great question. Her question was, does it say that you have to do rabbit pyrogen with the LAL, or do you run the some separate, the LAL all the time and the rabbit pyrogen just once, right? So there's two-part question we had to this. One, are they really saying that implants need to run LAL? That was the first question. The second question is, the LAL is a lot release test. So are you making implants, if you are making implants run the LAL, are you making it run it once, like a rabbit pyrogen is, or are you going to make them run it lot to lot? So as soon as we hear back from those two questions, we'll let you know, but we asked both of those questions. So The rabbit pyrogen will never be a lot-to-lot release test. It will only be a once. Well, I shouldn't say never because who knows. It shouldn't be in a lot. In fact, we're trying to get rid of the rabbit pyrogen someday, but the rabbit pyrogen should only be ran once unless there's a change. But the LAL, we're worried about maybe if we can do it just once, that'd be okay, but if we have to run it every lot, that's going to be a big impact. Good question. So this one, which I found interesting, because it says a rabbit pyrogen test need to be extracted at 50 degrees C regardless of contact duration. I don't know why they picked out the rabbit pyrogen to say regardless of contact duration, because if you remember before, it said to be ran at 50 degrees C if you have prolonged contact with the body. Well, I mean, if you don't have prolonged contact with the body and you want to run a rabbit pyrogen, you have to extract at 50 degrees C. It just seemed weird that they pulled rabbit pyrogen out of everything else. So we asked why the rabbit pyrogen test is pulled out. So we'll hear back from that. This one we actually fought because we disagree with. It said the mouse lymphoma test is preferred over chromosome aberration since it detects the broadest set of genotoxic mechanisms associated with carcinogenicity activity. We have a ton of data that shows the chromosome ab is actually more sensitive than mouse lymphoma. In fact, the chromosome ab could be considered the most sensitive genotoxicity test. So we submitted our data to the FDA and said, we think it's the other way around. I know that Japan submitted also data to show that chromosome ab picks up the majority of the genotoxic material. So we'll see what the FDA comes back with. This is the last one that we're going to be talking about today, and it's also one that kind of scares me, and that's deals with the chart. So I'm going to come to here. This is the chart out of the new draft, and in fact, if you guys want one of these afterwards, this is the chart in a slide rule. This is the G95 version, but they're the same. The only difference is, if you look, there are diamonds or circles that are considered alternative tests that may be considered. In the G95, it actually says that the diamonds are additional tests which the FDA considers may be applicable. So really what it means is if the reviewer thinks, I don't like your material or I'm scared of your material, they can ask for the diamonds, okay? The new verbiage down here is these additional evaluation tests should be addressed in the submission either by inclusion of the test or rationale of its omission. So they're now saying you either have to do the circles or justify why you didn't do them. Or before, we hardly ever ran them. So that was a little hard to pick. I don't know if many people caught that because it's the small words on the very bottom, which nobody reads in the legal documents. But we wanted to see if that really now what the FDA wants. Do they really want people to have to justify even the ones that the ISO, because the ISO doesn't have the circles. The ISO just has the X's. So are they saying that they want them to justify even ones the ISO aren't recommending? Okay, so that is the end of my presentation. So just so you know, remember, if you want one of these, they're up here. My cards are also up here. I also have some other information in the back. Are there any final questions? His question was, so he already has the device, and he already has Biocomp on it, but he's adding something to a coating, right? And that coating, that additive already has all the Biocomp information. Does he have to repeat his Biocomp? No, not necessarily. It depends on the information you have and the processing and everything like that. But it's a classic example of being able to use the justification to be able to justify out. You still have to do some Biocomp. We probably have to do some chemistry, some cytotoxicity to justify that. But this is one of those things where we would be able to justify out of biocompatibility based on the history and the processing involved of that coating. So we could take a look at it, but my experience is this is an example where we wouldn't have to repeat all of Biocomp. Good question. Great question. So her question is, I mentioned thrombogenicity is required. Is that the in vivo or the dog thrombo, or is it an in vitro alternative? So we have been successful in running a suite of in vitro alternatives. In fact, the in vivo thrombo has gone down in its running, and it's a horrible test. So you know, I can't even validate the in vivo thrombo because the control in one animal will have thrombus, and the negative control in the other animal won't have thrombus. So how do you validate a test that the negative control is different, right? It's just because the chemistry of the dog, just like us, changes from day to day. So one dog will have thrombus and one dog won't. It's not a very good test. But the FDA went through a small spell where they required it all the time. We're now, through the ISO and through a lot of work, we've been able to convince the FDA that it's not a good test. So we have been successful in running the in vitro thrombogenicity test, which is the PTT. Basically, all the in vitro tests are in vitro thrombo, because if you have complement activation, that can cause thrombogenicity. If you have hemolysis, that can cause thrombo. If you have platelet adhesion, that can cause thrombo. So we run the platelet adhesion, we run all the other in vitro tests, and as long as they come back fine, we justify that the animal test isn't needed because all those trigger. The only time that we can't get through that is if the geometry of the device worries the FDA. And so they'll run it to see the geometry makes sense. But we have been able to justify out of it with the in vitro. Very good question. Any other questions? Yes, no, this is great. I love talking about biocomp. We could do this all day. Yeah, how many people have ever seen the FDA I say it's a draft, but we're still following it? We see it all the time, right? So the funny thing is that first category are things we've already seen the FDA do for the last 5 years without having it written, right? So the new things, though, we haven't seen them enforce it yet. The ones in the possible changes, and so like the LAL, the chart, we haven't seen them enforce it yet. So until we get the new draft next year, hopefully next year, we won't really know. But right now, the possible changes have not been enforced. Good question. Any other? Okay, well, if you have any other additional questions for me afterwards, let me know. If not, thank you for your time. And for those of you that went all three, I'm sorry for having to